Hello, I'm Ben Miller. I'm from the University of Rochester, and I'm going to be telling you today about leveraging the AIM Photonics platform for new applications in biosensing and biomedical diagnostics. I want to start out by thanking the organizers for giving me the opportunity to speak to you today about the work that we've been doing in this area. And hopefully by the end of the talk, I'll have shown you some new ways that one can use photonic sensors for important biomedical applications. Now, I know many of you in this audience are more from the traditional photonics fields of data and telecommunications, but I think this will still be of interest, in particular because what we do in biosensing and diagnostics is leveraging work that's been done in the datacom and telecom fields to new market applications. Let me start by telling you a little bit what I mean by that. So again, most of you are focused on how we generate and work with large amounts of data, but where does that data really come from? You know, when you think about it, we live in an analog world, and sensors are the interface between the analog world and data. So anything that you're going to do to take information from the external environment and turn it into binary code that can be manipulated using a standard datacom model is going to rely on some kind of sensing device. Now, if you break this down into a couple of different areas, you can start thinking about the opportunities for markets. For example, in chemical sensing, this is forecast to be around 30.66 billion by 2024. Examples of how one can address chemical sensing include waveguide enhanced Raman spectroscopy. That's a very active area of research, including some work that we're doing along with collaborators at the Naval Research Lab. Not going to have time to talk through that today, but it's a very important application of photonic sensors. Today's talk focuses on biological sensing. This, using a similar market forecast, is predicted to be around a $76 billion market in the next couple of years. Depending on how these penetrate into the diagnostics market, that may be considerably higher. What we'll talk about primarily today are ring resonators and how those have been used in a microfluidics format to detect various biological entities. And of course, once you're able to do this, the opportunities for monitoring biological things, whether that's a protein in a human blood sample or a virus in a body of water, are many, and those in turn spawn many different types of scientific and market opportunities. So with that preamble, let's go ahead and get into the bulk of what I'll be talking about today. This work builds on a consortium that was developed through AIM Photonics to determine really what the opportunities were for sensors within the AIM Photonics offering, and to develop demonstration platforms of how sensors could be applied in useful chemical and biological contexts. This has been a really exciting group to work with. I won't read all the names, but it draws from various academic research groups, many industrial collaborators. I'll particularly note Orthoclinical Diagnostics, who's been a key partner for us in the biosensing and biomedical diagnostics arena, and companies like Synopsys, who's been crucial to our success in designing these new structures. By drawing from academia, Department of Defense labs, I already mentioned the Naval Research Lab, but we also had collaborators from the Army Chemical and Biological Research Consortium, and large and small industry. We were able to think about what the important problems were, what the important things were that we needed to demonstrate to show that AIM had a viable offering in this area. So in particular, for biological sensing, what we focused on are ring resonators. And for those of you who are perhaps familiar with ring resonators, but not so familiar with how these work in a biosensing context, what we really make use of is the fact that ring resonators have an evanescent tail beyond the waveguide. And if we expose that waveguide to the environment by producing a trench in the photonic sensor, that then sets up a situation where in the absence of the target molecule, we have a particular solution to the resonant condition for the ring resonator, and we get one resonant peak, or I should say a resonant peak every free spectral range of the resonator. And then if something sticks to that resonator, something that we want to detect, that changes the local effective refractive index, changes the solution to the resonant condition, and we get a shift that we can monitor. And importantly, this shift is dependent on the amount of material that sticks. And so it can be a quantitative measure of how much of that analyte is present. We can make this sensor specific to binding particular analytes using covalently attached molecules. And I'll tell you about that in just a little bit. So initially what we did was we developed designs for ring resonators using silicon nitride in the AIM Photonics platform. We modified the original multi-project wafer offering a bit in order to have a thicker bottom oxide that would give us the potential for using both TE and TM polarizations. There's been further work that's been done to look at different types of nitrides that I won't have a chance to get into today. But the end result is that we're able to produce silicon nitride ring resonators that have state-of-the-art performance. In terms of the Q factor, which can be as much as 10 to the fifth, and other characteristics that one monitors. And this is just an example of one of our chip designs where we did scan of coupling gaps between the ring and bus waveguides in order to optimize the extinction and that Q factor. And so now we're able to make these at 300 millimeter wafer scale with very high performance. We also optimized the trench process in collaboration with AIM so that we would have waveguides perfectly exposed to that external environment. So once we have these rings and we're able to make them reproducibly, let's go ahead and look at some biological targets. Here are just some initial examples of molecules that we've looked at. These are all proteins that are present in the human bloodstream, and their concentrations in a serum sample reflect particular disease states. So, for example, this molecule is S100B. It's a marker for a traumatic brain injury. If you get a TBI, whether that's a particularly hard hit on the football field, or if you are subject to an explosive device attack, some of this protein will leak out of your brain. And that concentration in the blood is a marker for TBI and an indication that you should be treated quickly. Human cardiac troponin I is this protein. That's something that is elevated in heart attack. So that's a reference diagnostic for heart attack. Two I'll talk about in a little bit more detail are C-reactive protein and interleukin-6. Both of which are proteins that are also elevated in myocardial infarction, but also in infection or other inflammatory states. And an interesting difference between these sort of highlights one of the challenges with detecting biological materials, in that C-reactive protein actually forms this pentameric structure, so very large structure. And in inflammation, it can be elevated to very high levels, so 0.5 milligram per milliliter, in some cases even higher. Interleukin-6 is a relatively small protein. In a healthy individual, baseline levels are down in the 5 picogram per milliliter regime. And in infection or inflammation, this can go up to around 250 picograms per mil. So you're looking at something that is more than 1,000-fold difference in concentration at the relevant disease states that we want to detect. And so that kind of shows the types of things that we need to be able to do with these sensors. So let's look at a little bit of data looking at trying to detect interleukin-6 and C-reactive protein. So to make these specific, what we do is we covalently attach an antibody to those proteins. One specific to IL-6 is attached to one ring. Another specific to C-reactive protein is attached to another ring, and that gives us two rings that can detect these two proteins. We can spot these using a system that is basically a fancy inkjet printer. It's a piezoelectric spotting system that we have in the University of Rochester Medical Center lab that I run, which is an outpost of the AIM Photonics test assembly and packaging facility. So this is really a manufacturing scale device that can spot picoliter droplet volumes on these chips. So here's one ring resonator sensor where we have rings arranged in two banks, and where initially in this case, this was spotted with an antibody to S100B. You can see that liquid there. An anti-C-reactive protein antibody on two different rings, anti-fluorescein as a negative control. And then we follow that up with something called stable coat that basically is a way of protecting the rings and gives it shelf stability. Here's another chip where we're doing that promised C-reactive protein and interleukin-6 experiment. So C-reactive protein antibody on one ring, control protein anti-fluorescein on another, and then interleukin-6, and then control protein anti-fluorescein on another. So these are all relative measurements, which means that we really don't even have to worry about temperature control for these experiments, although we do control the temperature of the solution. We then integrate the chip with a microfluidic system. So this is just a microfluidic cell that allows for fluid to come in and then out via these tubes, addressed via a fiber array with a standard 1550 nanometer tunable laser. And then we control the fluid flow using a computer-controlled interface that pushes fluid through the microfluidic system. So here's an example of what data looks like. This is flowing 100 nanograms per milliliter interleukin-6 in protein background. And what you see is that for this bank of two rings on one waveguide, you have very modest shifts for the control protein and for the anti-C-reactive protein. That's just what we call non-specific binding, so just stickiness. But then in this bank of two rings, you see a much larger shift for the interleukin-6 than the anti-fluorescein. And if you subtract this shift from this shift, you get the true change in resonance for this concentration of interleukin-6. We can reverse this and look at flowing anti-C-reactive protein. And here you see a big shift for anti-CRP, none or very little for interleukin-6. These systems are very well behaved analytically. You can look at different concentrations and get standard concentration curves. And we've been able to push these detection limits in a direct label-free assay below an nanogram per milliliter, or using a sandwich assay where we flow a sandwich antibody down into the femtogram per milliliter regime. So very high sensitivity assays. You can also get real-time information. So because we're scanning every second or two, you can look at the shifts as a function of time, and this allows you to get kinetic information about how these interactions occur, or as a way to monitor some system that you're interested in real time as you're flowing that biological material across the chip. So this is all great. From a research perspective, this is allowing us to do new things as research tools using these sensors. But what if we want to turn this into a diagnostic? How do we do that? The devices that I've shown you, the chips themselves are relatively inexpensive, although the chip cost obviously is a function of the real estate of the chip. You'll notice that what I've shown you so far doesn't have any other pieces of the sensing system integrated, although we are working on having integrated spectrometers and so on. That tends to increase the overall cost, as you might imagine. If we want something that's going to be useful as a clinical diagnostic, whether it's in a central laboratory or in the field, we now have to have something that's going to be on the order of pennies per test, and moreover, needs to be fast. So this was kind of where we were at the beginning of 2020, and as nobody needs reminding, the universe changed dramatically in early 2020 and provided us with an interesting and useful, important new opportunity in the diagnostic space. And specifically what I'm referring to is looking at detection systems for COVID-19. We decided to focus primarily on serologic testing because our sensors have been focused on detecting proteins. That's the opportunity to look at antibodies to COVID-19 and other viral diseases in a human blood sample. And so what this is useful for is looking at past exposure, meaning have you had COVID-19 in the past, but also as a way to look at understanding the human immune response to the virus, and as a way to look at vaccine efficacy. For example, I'm sure you've all been vaccinated, I've been vaccinated. How long does that immunity last? And as I'll show you, one of the things that we've been doing with these is looking at people who've been vaccinated with samples taken every month and seeing how vaccine antibody titers or concentrations change as a function of time. So the system we developed to do this, again, has to be cheap and fast. And what we call this is a disposable photonic system that would allow us to quantify and discriminate SARS-CoV-2 or COVID-19 immune response versus other coronaviruses and other upper respiratory infections. So here's this disposable photonics platform. What we did was we incorporated an exceptionally small photonic chip. This is 1 by 4 millimeters. I'll show it to you in a close-up in a minute, with a passive microfluidic system. This is a small plastic card that's about an inch on each side. And this incorporates a micropillar microfluidic system, basically little fingers of plastic that stick up. And then just by capillary action, or if you want to think about it, sort of like if you're familiar with pachinko machines, it always reminds me of a pachinko machine in terms of how these things stick up. But capillary action allows you to take a blood sample or a serum sample and have that flow passively under the chip and into a wicking zone that's basically just a sump for the sample. So the goal then was to be able to distinguish a positive sample from a negative sample. And I'm going to go over this fairly quickly, but this was published recently in Lab on a Chip if you want to know more. So here's what the photonic sensor looks like. Again, a grain of rice sized. So we limit the cost in terms of the photonic sensor chip itself. We use grating couplers to get light in and out of the sensor chip. And then this particular version has 2 rings, one for the experimental detection area and another as a control. We've now expanded this using the same footprint to an 8-plex, so 8 rings on multiple waveguides. That allows you to manufacture these at very large scale, getting tens of thousands of chips from a single 300 millimeter wafer. And we then also developed an optical read head system that allows us to interface with a lensing system that takes the laser light, sends it vertically into the grating coupler on the PIC, and then retrieves light out the other grating coupler to send to a standard detector. So here's a close-up of what this micropillar fluidics card looks like. Again, we've got a sample introduction zone that flows underneath where the PIC goes and then on into a wicking zone. Initially, we made these using a hot embossing process developed by Nate Cady and his team at SUNY Polytechnic. More recently, we've also been doing this using injection molding. Here's a close-up of what these micropillars look like. And again, you can see they're just fingers of plastic that stick up into this zone and pull fluid under the PIC. So to detect antibody responses to SARS-CoV-2, we have primarily focused on the spike protein in different domains. The spike protein is what gives coronaviruses their name. They form a crown around this viral capsid, and they interact with the ACE2 receptor on human cells. And so we took two different versions or subdomains of this spike protein, the S1 plus S2 or RBD subdomain, and attached those to the chip, again, using the same piezoelectric spotting system. Again, we use antifluorescein as our control, and then follow that up with stable coat so that these can be shelf stable for months, if not years. So the workflow, initially we took blood samples, separated out the serum, and applied that to the consumable. So we're looking for an initial spectrum that has our SARS-CoV-2 antigen, that protein, and an antibody. We look for a shift if antibodies bind, and we can follow that up if we want to confirm it's an IgG response with another protein. So here's what a positive sample looks like. You can see a strong shift even at a minute. A negative sample, basically no shift when subtracted from the control. And if we look at a series of people with COVID-19, we can easily discriminate those from those who had not had COVID-19. Now, as people began to get vaccinated, we looked at those as well. And I should say we also looked at the S1 plus S2 antigen, I should say, and that also works quite well. With the vaccinated samples, what we see is that we can determine development of anti-COVID-19 antibodies after vaccination. And we can also watch this trail off over the course of a few months. So this is turning out to be a really useful, very rapid, inexpensive photonic sensor tool for looking at COVID diagnostics and vaccine monitoring. It's reproducible. We've been able to show that works quite well in replicates of the same sample, and we're continuing to work on the sensitivity of this system. We've also used this for looking at detection of the spike protein itself following vaccination. And I'm not going to have time to go into this in a lot of detail, but it's an interesting question. After you get vaccinated, how much of this protein is actually there, and how long does it stick around? So what we did here was, rather than putting down the SARS-CoV-2 or COVID-19 protein itself and detecting antibodies, we reversed things, sort of going back to what I showed you with the C-reactive protein and interleukin-6 detection, where we put down an antibody to the spike protein and detected that. So here's what that looks like: deposition of the antibody, covalent attachment to the surface, and this gives you a little bit of a sense of the chemistry that we actually use on the surface of these photonic sensors. Here we use bovine serum albumin as a control. And again, the same sort of optical read head system. Now, these are using the injection molded structures rather than hot embossed structures. So again, we're looking at a shift in the anti-RBD relative to bovine serum albumin, the control. And so after developing a calibration curve that let us know what the concentrations were as a function of relative shift, we looked at a series of subjects immediately post-vaccination and pre-vaccination. What you see is that pre-vaccination, they don't have any S1 plus S2 protein present. Immediately after vaccination, you can detect a shift for the presence of that protein produced by, in this case, the Pfizer messenger RNA vaccine. And so with this set of samples, you can see that the spike protein is elevated and then rapidly declines. In most of these subjects, this person actually had a coronavirus infection. We're not exactly sure whether it was COVID-19 or a related one that gave a signal still out at a longer time point. But you can see that really within a couple of weeks, the spike protein produced by the messenger RNA is gone, while the antibody titer goes up. So that shows the versatility of this system. And then just briefly, what about other sensor types? One type of sensor that we worked on quite a bit in the lab is 2D photonic crystals. These are ultra small, potential for single molecule sensitivity. And I'll just close by saying we've now shown that we can make these at wafer scale on the AIM Photonics platform, also using silicon nitride. These devices have very high fidelity to our designs, again, designed using OptoDesigner. But the cool thing here is that it shows that using a foundry, we can make things that have sub-wavelength dimensions. And these actually do work well as sensors, and we're continuing to examine them. So to conclude, this disposable photonics system provides a very rapid method for detecting antibodies to SARS-CoV-2. It's something that's compatible with high volume manufacturing, so very manufacturable, extendable to other pathogens, and I think highlights what one can do with the AIM Photonics platform for sensor fabrication, even in including things like making sub-wavelength structures for 2D photonic crystals. So with that, I'll just thank the folks who've done this work. I've shown you the work of a number of my students here, particularly Michael Bryan, Dan Steiner, John Cognetti, Joe Buchkowski isn't in this picture, but another one who very critical to this effort, Shurik Zavriev, and others who've been active here. Also Ethan Luda, Alana Close, and other members of my group. I've had a terrific set of collaborators from the AIM Photonics sensor team. And then of course, particularly want to thank my funding sources in this SARS-CoV-2 work that was supported by the Department of Defense, and they've been terrific partners as we've developed this. And I'll thank you for your attention, and I'll be happy to answer any questions that you might have. Thank you.